those of you that have joined us thus far. This is Marie Latulippe with ILSI North America, and this is a webinar organized by the Gut Microbiome Committee of ILSI North America, and we're putting these webinars on as part of our efforts to understand the links between diet, gut microbiome, and health. So it's part of a series. And today we have Dr. Gerard Clarke joining us, who is a lecturer in the Department of Psychiatry and Neurobehavioral Sciences at the University College Cork. Gerard graduated with a BSc in chemistry from the National University of Ireland, Galway, and that was followed by a master's in neuropharmacology from the same institution. And then after that, he received a PhD in the Department of Psychiatry from University College Cork. Gerard was then a visiting scientist at the University of Mississippi Medical Center in Jackson before working with several multinational pharmaceutical companies, and those included Wyeth, Pfizer, and 3M, in addition to other postdoctoral experiences. So Dr. Clarke's current research focus takes a translational approach to the assessment of neuropharmacological indices of stress-related disorders, such as depression and irritable bowel syndrome. And this approach incorporates studies in both clinical populations and animal models using an early life stress template that produces a specific disease phenotype in adulthood. He's particularly interested in advancing our understanding of how tryptophan degradation influences psychopathology. His research also looks at the impact of microbiota manipulation on the central nervous system, and he'll tell us a bit about this work today. His research has been recognized internationally, and Dr. Clarke is also currently a PI on a Health Research Board funded study, that's a state agency of Ireland, and that looks at the links between the kynurenine pathway metabolites, which is a pathway of tryptophan metabolism, and cognitive dysfunction in IBS, as well as other work that's funded by the Health Research Board. So with that, I will turn it over to you, Dr. Clarke. Hopefully everybody can see your slides. I can, and I can hear you pretty clearly. So take it away.
Okay, well, thanks very much, first of all, for the kind invitation and the opportunity to talk about some of my research in this webinar. So, as you said, I'm based in the Department of Psychiatry and Neurobehavioural Science here in University College Cork, but also APC Microbiome Ireland. And APC Microbiome Ireland is a Science Foundation Ireland funded research centre, and we're really dedicated to the study of host microbe interactions. So we do that across a number of research teams, and also engage quite a lot with industry to look at specific projects and kind of answer specific questions about the gut microbiome and health. So within the research teams that we have in APC Microbiome Ireland, I'm kind of focused on the brain-gut axis section. And I suppose what we've been thinking about really is that traditionally, when we've thought about treating stress-related psychiatric disorders, quite logically, you know, we've aimed our treatments at the organ which is located above the neck. But a lot of our research suggests that maybe we should be thinking a little bit about what's going on below the neck as well, and that maybe we can target our gastrointestinal microbiota to produce benefits at the level of the central nervous system. So really what we're about is, can we move the goalpost a little bit in the treatment of stress-related psychiatric disorders? And I suppose the question I posed at the top of the slide isn't really true anymore, and that the gut microbiota is no longer, you could say that it's forgotten, but certainly for a long time, until the current surge in interest over the last kind of 15 years, I suppose, it was considered pretty benign in a way. And it's kind of relatively recently that we've become to appreciate its real importance. And that includes from a range of different perspectives, including some of the functions that it has in terms of protective functions, structural functions, in terms of regulating the barrier in the gastrointestinal tract, and a range of other metabolic functions which are essential and which the host could not do without. But I suppose it's only more recently still that we've appreciated that although we have this gastrointestinal tract which is teeming with these microorganisms and that they have an important role to play in health and disease, that they also might have an impact at the level of the central nervous system. And that's kind of given rise to this concept of maybe mind-altering microbes. And a lot of the work that we've done to date shows that these microbes could have an impact on things that are relevant to psychiatry, like anxiety, depression, stress, pain, and cognition. Now, Milo is very interested in the mechanism that might be underpinning this, particularly the role of microbial regulation of tryptophan availability and metabolism, because it overlaps quite a lot with the behaviors which are influenced by the gut microbiota. So what I'm going to do for the rest of the seminar, really, or the rest of the webinar, is to give you a broad overview of this area, and then try and come back towards the end, if I have some time, to talk about the translational implications and the opportunities that arise from these observations for intervention to improve brain function and behavior. So I suppose the model that we work off is that the gut microbiota can recruit this brain-gut axis, which is this two-way communication system between the brain and the gut to produce effects at higher brain centers. So, excuse me. So, if we have a healthy gut microbiota, and at least in adulthood, what we mean by a healthy gut microbiota is one that's both stable and diverse, then that kind of contributes to a normal gut physiology. You get appropriate signaling along this brain-gut axis, and that confers a healthy status at the level of the central nervous system. Now, if something goes wrong with your gut microbiota, and I guess what we've seen to date in that most disease states, what tends to go wrong, for example, is that the diversity might narrow, or maybe the gut microbiota becomes less stable in certain disease states, and then that might contribute to abnormal gut function, leading to inappropriate signaling along this brain-gut axis, and manifesting as alterations in behavior at the level of the central nervous system. Now, it's important to appreciate as well that this is a two-way street. So stress, which is perceived at the level of the central nervous system, can also signal down to your gastrointestinal tract and influence the composition, structure, and function of your gut microbes. Now, in terms of how all this works, well, as I said, the microbiota probably recruits the scaffolding provided by this brain-gut axis, and the brain-gut axis consists of a number of different components. So, for example, your gastrointestinal tract is very heavily innervated by the vagus nerve, and that's a direct highway of communication between the gut and the brain. But there's also neuroendocrine and neuroimmune routes involving cortisol or immune factors like cytokines, for example, through which this communication can occur. I'll come back a little bit later in the webinar to talk about microbial regulation of tryptophan metabolism. But for now, it's important to bear in mind as well that these microbes in your gastrointestinal tract are really like mini factories. So they're constantly churning out microbial metabolites from the foods that you eat, for example, short-chain fatty acids from the fiber. And these microbial metabolites can impact on host physiology. Now, in terms of where we get our microbiota from, well, in utero, your gastrointestinal tract is pretty much a blank canvas, really. So then you see the first kind of brush strokes applied during the birthing process. So you get your microbes initially from your mom, and that leads then to this trajectory until about maybe 3 years of age, at which point the microbiota becomes somewhat resembling the adult-like microbiota composition that we would probably know most about. So there's this chaotic period at the start of life during which the gut microbiota assembles before it reaches its adult-like status. And you and I probably have very different microbiotas, because there's a lot of different factors surrounding our lifestyle and stress exposures, for example, which would help define the compositional characteristics of your gut microbiome. So as I said, there are stable and chaotic periods. So as you age, this tends to lead to changes in your gut microbiota. We know that this initial trajectory during which your microbiota is assembled following birth can be influenced, for example, by your mode of delivery. So if you're born paravaginum or born by C-section, that will initially lead to a different initial trajectory as you move towards the adult-like composition at about 3 years of age. So maybe if you're born between those two different modes of delivery, you might eventually catch up to each other. but your initial phase of microbiome assembly will be different. Then there are different things like host genetics, exercise, stress exposures, medication use like antibiotics maybe, that will lead to alterations and differences between an individual's or two individuals' microbiota. So I suppose the key thing there is that we have this second genome really, which is modifiable because we can change it with diet, for example, which is an important way in which the microbiome can be sculpted. And perhaps an extreme example of that can be seen in this study from Tim Spector, where he sent his son to McDonald's for 10 days in a row. And that really narrowed the diversity and composition of his son's gut microbiota. So we can also use this, of course, beneficially by increasing the diversity of our diet to promote a healthy microbiota. And a lot of issues may arise, I suppose, because in the Western world and with the Western diet, we maybe are a little bit deficient in the recommended fiber intake, for example, and that can be have implications for the health status of our gut microbiota. So how do we know all this? Well, a lot of what has come to the fore over the last while, I suppose, has been down to the improvement in the technologies that are available to look at the gut microbes. So one of the advances really has been in sequencing, and that allows us to conduct a census of the gut microbes to find out who's there and in what sort of abundance. So for example, you can get a stool sample from a study participant, extract the microbial DNA from that sample, and then run it through these sequencing pipelines to get an idea of the diversity, and also then drill down into the data a little bit to kind of find out different compositional characteristics and look at maybe ideally even in ideal study outcome, you might find the missing microbes between one disease state and another. And I suppose these sequencing approaches have told us a lot, and we know a lot now about the different stages of gut microbiota assembly, and also different periods in life during which the composition might change. So we know, as I've mentioned, that there are chaotic periods early in life before the adult-like gut microbiota emerges. We know as well that as you age, maybe the diversity narrows a bit. Even during pregnancy, there seems to be somewhat of a remodeling of the gut microbiota, with the idea that maybe this is intended to pass on the optimal gut microbiota initially during the birthing process. Now, to try and get at the kind of questions that we've been asking about the gut microbiota in terms of how it might influence brain function and behavior, you can use a number of different approaches. I suppose some of these are pretty intuitive and don't need much explanation, that you can administer probiotics or antibiotics to perturb the gut microbiota and see what the consequences of that are. And an approach that we use quite a lot, which maybe merits some more elaboration, is the use of germ-free animals. So I'll go on to that in a minute, but collectively, I suppose what these different approaches have told us is that the behaviors which are maybe under the influence of the gut microbiota relate to things like anxiety, depression, pain, and cognition. So the use of germ-free animals then has been very kind of instrumental in charting out the kind of things that are influenced at higher brain centers by the microbes which are resident in your gastrointestinal tract. Now, the use of germ-free animals is not a new approach. It's been used quite a lot. I like this figure showing the old isolators that were used, and particularly this picture, as they've taken the trouble even to label up the technician in the diagram. But it does illustrate that it's an old approach. It's been used quite a lot in immunology, and it's only that relatively recently it's been applied to the type of questions that we've been asking. And there are some caveats, of course, that come with its use. So we know that there's probably no real clinical, translational clinical equivalent where there's such a complete obliteration of your gut microbiota across your lifespan. And you know, one of the extreme examples might be the boy in the bubble, but outside of that, it's hard to come up with a direct clinical equivalent. But nevertheless, it's a really neat approach because you can compare a conventional animal to a germ-free animal, and also then manipulate the germ-free animal, so you can add a microbiota back in, or you can mono-associate the germ-free animal with one particular bacterial strain. See what are particular aspects of brain function and behavior that you've noted to be altered, might be rescued by colonizing the animals or monoassociating the animals with a particular bacterial strain at different stages in life. Now, the things that are influenced by the gut microbiota range across a number of aspects of host physiology and behavior, and I guess one of the things that turned up most robustly is the impact of the gut microbiota on our stress response. Now, I guess we're all familiar with the different stresses that we encounter in modern life. We have some of the issues maybe with the Trump presidency, and over here at the minute our minds are very focused on Brexit and what the consequences of that are going to be for Ireland in particular. And I suppose we all encounter different stressors on the roller coaster of life, and we all probably react a little bit differently to those stressors. And really, that's the key point, I suppose, is that it's not the stress that kills us per se, it's how we react to it. And in terms of how we react to stress from a biological perspective, at least, the microbiota seems to play an important role. So one of the indices or one of the readouts that we use to measure the stress response would be the production of stress hormones like cortisol in humans or corticosterone in rodents following different stress exposures. And this corticosterone production reflects activation of the hypothalamic pituitary adrenal axis, this HPA axis. So what we know from the germ-free animals is that if you stress out a germ-free animal, that it will have an exaggerated stress response, and that's indicated by this increased production of corticosterone in germ-free animals compared to those which have a conventional microbiota. And that ties in pretty well with what we know from studies following probiotic administration, where particular putative probiotic strains, like in this case Lactobacillus rhamnosus, can reduce or blunt the stress-induced production of corticosterone, which would be considered beneficial for a lot of stress-related disorders. Now, I teach a lot of the medical students here in UCC, and I tell them about the neurobiology of things like depression and other psychiatric disorders. And I suppose one of the things that I emphasize there is that there are specific brain regions which can be linked to specific functions. And even though that's a little bit maybe overly simplistic and there are a lot of important interconnections between these brain regions, it's still a pretty useful way of thinking about things. So when we think about the hippocampus, for example, that's a brain region that's linked to learning and memory. When we think about the amygdala, that would be linked, for example, to fear and anxiety. While when we think about the areas of the frontal cortex and the prefrontal cortex in particular, that would be very important in the regulation and integration of emotional experiences. And all of these brain regions seem to be markedly different as a consequence of growing up germ-free. Now, one of the nice things about this, although there's obviously differences in the brain structure between the human brain and the rodent brain, still some of these brain regions are present and subserve the same functions, particularly the hippocampus, the amygdala, and areas of the frontal cortex, which is why this is a translationally relevant approach. So if we look, for example, at the amygdala, at a structural level, even that is different in these germ-free animals compared to conventional controls. So the volume of the amygdala, the size of the amygdala, and particular nuclei within the amygdala is different in germ-free animals. You can see the diagram there, the difference even at the neuronal structural level, where germ-free animals have these much more elaborately branched neurons compared to the control animals. And this has functional implications. So there are specific behavioral assays that you can do. The one depicted on this slide here is known as cued fear conditioning. And we know that that specifically engages this amygdala brain structure. And germ-free animals respond differently during this behavioral assay compared to conventional animals. So they have difficulty, for example, with fear recall. And that indicates that these anatomical alterations, I suppose, that we see in the amygdala probably have functional implications. And indeed, if you look at a lot of what, behaviorally speaking, goes wrong in these germ-free animals, in terms not just of anxiety and fear, but also in terms of pain and social behavior, then a lot of that maybe can be tied back to these alterations in this amygdala and the functional consequences of that. Another region that I mentioned has been important is the prefrontal cortex. And what you can see on this slide here is that there's hypermyelination of the nerves in the brain of germ-free animals compared to conventional controls. Now, myelin is kind of the sheath that wraps around these nerve cells, I suppose, and is important in the conduction of signals. So you can see in the bottom there that the germ-free animals have a very much thicker myelin sheath compared to the control animals. And at a transcriptional level in the genes that are important for the regulation of myelination, we can also see that germ-free animals have excess of gene expression for these important genes. Another important brain region is the hippocampus. I identified it as being important for memory and learning. And we see that BDNF in the hippocampus is reduced in the germ-free state. Now, BDNF stands for brain-derived neurotrophic factor. It's very important for neuronal survival and growth. It's like a fertilizer. You can think of it like Miracle-Gro for neurons. And we know that stress will reduce BDNF, for example, and also that antidepressant treatment may in fact ultimately work by increasing BDNF levels. So just going back to signaling along the brain-gut axis, and we mentioned before the different mechanisms that might be at play here. So maybe we can zone in now on tryptophan metabolism. So tryptophan, as you might know, is this amino acid. It's an essential amino acid. It has to be supplied in the diet. And once it's available in the diet, then it's available as a precursor for a number of very important neuroactive compounds. So it probably would be best known as a precursor to serotonin, and that's because serotonin is the target for antidepressant drugs. But serotonin also has a very important role to play in the gastrointestinal tract. So even though it has very important behavioral roles in the central nervous system in a number of aspects of brain function and behavior, in the gastrointestinal tract, it's essential for normal function like gastrointestinal secretion and motility. And actually, the largest reservoir of serotonin in the human body is in the gastrointestinal tract, and in the enteric nervous system you have important receptors like the 5-HT3 and 5-HT4 receptor subtype, which are expressed there and can be activated by local serotonin production. Now again, going back to the hippocampus and germ-free animals, we can see that if you grow up germ-free, you have higher levels of circulating tryptophan compared to conventional animals. This is something that can be rescued by colonization of the animals post-weaning, and is probably related to the way the microbiota regulates the metabolism of tryptophan along a different pathway, which I'm not going to go into today, but for your information is known as the kynurenine pathway. The consequence in the central nervous system of this alteration in circulating tryptophan concentrations can be seen in terms of elevations in serotonin and its main metabolite, 5-HIAA, in the hippocampus of these germ-free animals. And interestingly, this increased production of serotonin in the hippocampus and the central nervous system was not something that could be rescued by colonization of the animals post-weaning, so it seemed to be hardwired in there. Another interesting feature of this research, I suppose, is that a lot of the changes that we see in the germ-free state manifest in male animals, but not in female ones. So we can see that although male animals have increases in serotonin and its metabolite in the hippocampus, in the male animals, it's not present in the females. And again, I don't have time today to go into this in detail, but of course the microbial metabolites which are produced in the gastrointestinal tract play a very important role in regulating host serotonin biosynthesis locally there. So for example, short-chain fatty acids produced by the gut microbiota can stimulate the enzymatic activity of tryptophan hydroxylase, which is responsible for the production of serotonin in the intrachromatin cells in your gastrointestinal tract. Excuse me. So I suppose just a summary of this part of the talk, I suppose, is that the gut microbiota regulates a lot of behaviors relevant to stress-related psychiatric disorders. So we know it's important in behaviors like anxiety, depression, stress, and cognition. And there's a big overlap between those behaviors and the fact that tryptophan and its metabolites can influence these behaviors, and there is a microbial evidence for microbial regulation of tryptophan metabolism. So a lot of what we're seeing with the ability of our gut microbes to influence brain function and behavior could well be down to their capacity to modulate tryptophan metabolism and availability. And even though I've emphasized microbial regulation of host tryptophan metabolism here, it's important as well to bear in mind that the microbes themselves have a lot of enzymes which are capable of metabolizing tryptophan, potentially converting it into metabolites like indoles, which can also impact on host physiology. So I suppose in a way where we are in the field at the minute is that there's a really promising body of preclinical research, and what we really need to see now is whether this preclinical research is going to effectively translate to the clinic. So can we bring this from bench to bedside? And I suppose there are always challenges in bringing animal research into the clinic, some which are generic to all animal studies, but also some specific challenges associated with the microbiota. So that's what we've been kind of interested in, to see whether what we've seen preclinically is also apparent in the clinic. And one of the things that we've looked at is whether there might be an altered gut microbiota in subjects with depression. So for example, when we applied these sequencing approaches to the study of the microbiota in depression, we've seen that in common with a lot of other disorders, there is reduced microbial diversity associated with depressive-like behavior and in clinical depression. And we can see, if you drill down into the data a little bit further, for example, that Prevotella, which is a genus of gram-negative bacteria, is reduced in depression compared to healthy controls. And that kind of stood out to us a little bit because Prevotella, an increased abundance of Prevotella, is very often associated with the Mediterranean diet, which may have a protective role in depression. Now, one of the fascinating areas or aspects of research in this area is the transplantation of behavior via the gut microbiota. So this was an early study from McMaster, from Birchick and Collins, where they essentially swapped the different behavioral profiles of these two mice just by transferring the microbiota. So the field really is taking advantage of that then to see whether disease-specific or disease-associated microbiotas might have a causal role in producing relevant behaviors, because a lot of the studies, and the one I just mentioned there, is cross-sectional in design. So although we can see that there's an alteration in the gut microbiota at that time point between depressed subjects and controls, we can't really tell from that whether the microbiota alterations might be important for producing specific behavioral alterations. So, as I said, the field has adopted this microbiota transplantation technique to see whether the disease-associated microbiotas might have behavioral consequences. So indeed, what we've seen was when we transferred the microbiota from subjects with depression into these into our animal models, that we're able to transfer some of the behavioral phenotypes associated with depression as well. So, for example, anhedonia-like behaviors, the inability to experience pleasure and normally pleasurable things, was transferred via the gut microbiota. The anxiety, which is very frequently comorbid with depression, was also transferred via the gut microbiota. And even at the neurobiological level, we could see that when we transferred the microbiota from depressed subjects into our animals, that they had alterations in the metabolism of tryptophan along this specific pathway, which is thought to be a neurobiological marker of depression. And again, as time has gone on, I suppose a lot of the initial studies in this area were somewhat smaller populations of studies. Very recently, we've seen this important study from Jeroen Rees and colleagues in Belgium, which is the largest study to date, I suppose, associating the microbiota with depression and indeed quality of life. And what was very important, I suppose, about this study was that, you know, it was pretty large, and that they initially looked at over 1,000 people and this cohort study they had, and followed up by validating it in a different population. So really kind of robust evidence that the microbiota might be altered in depression and linked to quality of life. Now, another clinical disorder that we study because of its links to the microbiota is irritable bowel syndrome. That's a functional gastrointestinal disorder. It's characterized, I suppose, by abdominal pain, constipation, and diarrhea, and the abdominal pain may be underpinned by visceral hypersensitivity in the gastrointestinal tract. We were initially interested in it because there's a lot of psychiatric comorbidity in irritable bowel syndrome, in terms of anxiety and depression. And I suppose in many people's eyes, it can be viewed as this prototypical microbiome-gut-brain axis disorder, which is stress responsive. Now, again, we can see if you look at the microbiota, which is associated with irritable bowel syndrome, that transferring it to animals can recapitulate some of those behaviors in the animal models. But what was really interesting about this study was that they segregated their patients into subjects who had pure IBS, I suppose, if you like, but then also irritable bowel syndrome with comorbid anxiety, and they were able to transfer the distinct behavioral profiles via the microbiota. So if you had just IBS, it was only the IBS characteristics which were transferred via the microbiota, whereas if you had IBS and comorbid anxiety, then you also transferred the gastrointestinal features, but also the anxiety features. So that only happened if you had the comorbid anxiety expressed. Now again, and I'm not going to go into too much detail in these studies, but because of what we've noted to be altered in our preclinical studies, we were interested in whether, for example, subjects with IBS might exhibit alterations in cognitive performance. That's what you can see here. This is a computerized touchscreen battery of tests known as the CANTAB battery. This particular test engages the hippocampus, as known as the paired associated learning test, and it tests visuospatial episodic memory. So what you can see, I suppose, in the subjects with IBS compared to healthy controls is that they make more errors. This is something we followed up longitudinally, looking 6 and 12 months later as well, and showed this to be a persistent, albeit low-grade aberration in performance in this particular test. Again, because of our knowledge that the microbiota has an important role to play in the stress response, we reasoned that subjects with IBS should show some alterations in this following an acute stress challenge as well. And I've talked before about the acute stress response of the germ-free animals compared to healthy controls. Well, we can also look at that in a clinical situation. And this particular behavioral test here is known as the Trier social stress test. So it involves a number of different procedural stages, I suppose, but the key point is that following this acute stress protocol, you get robust activation of this hypothalamic pituitary adrenal axis. So you can measure that in terms of cortisol production in clinical subjects. And indeed, what we've seen when we acutely stressed these subjects with IBS was that although the magnitude of the response in terms of cortisol production was similar, which can be seen in the top here, subjects with IBS had difficulty in turning off cortisol production, which may have important implications for their symptomatic profile. And indeed, if you look at other aspects which we've noted to be altered in germ-free animals or following different microbiota manipulations, you can see, for example, that there's alterations in tryptophan metabolism in subjects with IBS as well. So you have increased metabolism of tryptophan along alternate pathways and the production of serotonin. And indeed, that ties in with what we know about certain strains of probiotic, which are beneficial for looking at some symptoms in IBS. And we know from our preclinical studies that this bifenfanta strain, for example, was able to modulate the metabolism of tryptophan in our preclinical models. So I suppose knowing all this, the question arises as whether we can beneficially manipulate the microbiota in human subjects to try and produce benefits for brain function and behavior. And one of the terms that my colleague, Professor Dinan here in the Department of Psychiatry has coined is that of a psychobiotic. So that was initially a probiotic that might have a mental health benefit, but it's also been expanded now, I suppose, to include many other microbiota-directed manipulations, including, for example, prebiotics, which might be used to promote the growth of beneficial bacteria. So we were talking about maybe a psychobiotic revolution, whereby instead of targeting the brain to treat psychiatric disorders, that maybe we might be able to harness the power of the microbiota to produce benefits as well. So a lot of what we've been focused on, I suppose, is screening in our preclinical assays for candidate psychobiotics that might have benefits in when applied to clinical populations. So we use a variety of behavioural screens that capture things like anxiety, depression, cognition behaviours, as well as aspects of gastrointestinal function to try and pick up in our preclinical screens a psychobiotic that we could move then into testing in human populations. And a lot of these tests that we use will be very widely used in the pharmaceutical industry to assess antidepressant potential and anxiolytic potential of new drugs. So things like the forced swim test or the open field have really come, I suppose, from the pharmaceutical industry. So what we noted, I suppose, is that one particular strain stood out to us in our preclinical testing. That was this biflongum strain, which produced benefits here in terms of cognitive performance on a particular maze, as well as producing benefits on aspects of stress-related behaviour and physiology in our animal models. So when we moved this into our healthy human subjects, we were able to show that following consumption of this psychobiotic or this candidate psychobiotic for a month, subjects reported less stress on the perceived stress scale. When we stressed them out, this time using a socially evaluated cold pressor test. So this is another acute stress challenge that we use. This time participants' hands, they're submerged in an ice cold container of water. They are told that the procedure is being recorded and scored, will be scored later on, and it's usually the instructions are given by a member of the opposite sex. And combined, I suppose, all these things reliably induce an activation of the HPA axis, which can be picked up using cortisol readouts. And what we can see from our psychobiotic in this particular test was that the cortisol output was reduced under the psychobiotic condition compared to the placebo arm in the study. And we can see as well, we measured brain activity using EEG and also measured performance on the paired associated learning cognition test, which is the test I mentioned earlier as being abnormal in subjects with IBS. So we can see that the psychobiotic administration is also linked with alterations in brain activity and marginally superior performance on this test of cognition. More recently, some colleagues in Germany went on to use this with a different brain imaging approach. So they used this biflongum strain in, again, a social stress scenario. So this is a cyberball game, and it involves elements of exclusion, which are found to reliably activate the stress response again. And again, this same candidate psychobiotic was able to modulate resting brain activity and brain responses measured during this social stressor. So we thought this was pretty good. It was pretty interesting. So we went back to try and screen some of our other candidate psychobiotics. In particular, this Lactobacillus rhamnosus JB1. Now this, in our preclinical tests, had one of the strongest preclinical profiles in terms of beneficial regulation of aspects of the stress response and cognitive performance, but it did absolutely nothing when we repeated the protocol that I showed you previously with this different strain of bacteria. So I suppose this emphasizes a few things. One, I suppose that we can't generalize in terms of the impact that a psychobiotic or a probiotic strain might have from one strain to another, that the effects are very strain specific. And also, I suppose, it illustrates some of the challenges in bringing very promising preclinical research into the clinical arena. So I suppose one of the things that we need to take on board maybe is that we may look to modify our pipelines for psychobiotic discovery a little bit. And indeed, we know now a lot more, I suppose, about the aspects of brain function and behavior and the potential mediators that are at play to try and identify psychobiotics that might have desirable properties in terms of their capacity to regulate the immune response or to modify intestinal permeability or to regulate host tryptophan metabolism that might lead to benefits in terms of brain function and behavior. So as I said, we originally thought of psychobiotics in terms of probiotics with mental health benefits, but that's since expanded to think about other things like prebiotics, which might promote the growth of beneficial bacteria. So we can see one example here again from a preclinical study showing that this Foss and Goss prebiotic was able to reverse the consequences of chronic stress. So chronically stressing animals in this preclinical protocol can induce alterations in social behavior and cognition, and can induce anxiety-like behaviors. And all of those consequences can be reversed by administration of this prebiotic. Now that ties in pretty well, I suppose, with what we know from earlier studies and clinical samples as well. This is an example from Phil Burnett's group, where he was able to show that prebiotic intake was able to reduce the cortisol response that occurs following waking. And another really interesting angle here is how we might envisage using these microbiota manipulations. So this is a study from Peter Bork, where he showed that there's an extensive impact, I suppose, of host-directed drugs on microbial activity and growth. So a lot of drugs that he evaluated, which were human-targeted essentially, would also have antimicrobial activity. And drugs which are very commonly used in psychiatry, like antipsychotics, which would be used to treat schizophrenia, for example, seem to have antimicrobial properties as a class. Now, this is interesting to us because one of the problems associated with antipsychotic use is that they're linked with weight gain quite a lot. So it's a very bad or detrimental side effect associated with antipsychotic use, which is very problematic in the clinic. And work from Siobhan O'Mahony here in Cork has shown that this weight gain might be linked to the effects of antipsychotics like olanzapine on the gut microbiota. So for example, if you administer antibiotics or you administer olanzapine to germ-free animals, you don't see the same weight gain, heavily implicating the microbiota in this aspect and this detrimental side effect of antipsychotic use. And indeed, this is another context in which prebiotic manipulation might be considered. And we see again this study from Phil Burnett showing that they were able to administer prebiotics and attenuate the induction of weight gain following olanzapine use. Now, of course, as I mentioned, diet plays a very important role in sculpting the microbiome. So a lot of these findings come at a very interesting juncture in psychiatry when work from Felice Jacka in Australia, for example, is been instrumental in promoting the idea that nutritional medicine might play an important role in the improvement of psychiatric conditions. So, for example, in her SMILES trial, which was published a couple of years ago, she was able to show that the adjunct treatment, I suppose, with a Mediterranean-style diet was able to improve depression scores in subjects with depression. Now, I suppose the elephant in the room here that has to be mentioned in all of these talks is the fecal microbiota transplantation story. So this, I suppose, has shown a lot of utility in the treatment of recurrent Clostridium difficile infection, and there's a lot of interest in where it might go from there. So we have seen, for example, it dated back to the yellow soup that was used in Chinese times to, as I said, the effective treatment of Clostridium difficile infection now, and the possibility that might be used for the treatment of other conditions that are linked to the brain-gut axis. So I don't know about you, I think I'd rather be a donor than a recipient in these types of studies. But I suppose if you look around and you do need a donor, you want to be very sure that they are of a sound psychological profile to make sure that aberrant behaviors aren't transferred when you do get your stool donation. And joking aside, I suppose, if you look at some of the guidelines that have come out recently from Europe on the use of fecal microbiota transplantation in clinical practice for the treatment of recurrent Clostridium difficile infection, they do indeed advocate screening to rule out a history of psychiatric disorders before the stool can be considered acceptable for transplant. So I suppose our perspective here at the APC is that the gut microbiota plays an important role in regulating behaviors which are important for good mental health, and that maybe we can mine for or target it with psychobiotics, which would include things like probiotics, prebiotics, dietary manipulations, maybe down the line fecal transplants, or The more palatable version, I suppose, would be defined microbial consortia that might produce the same benefits via a more pleasant route of administration. And the other perspective, I suppose, is an area that's a hot topic right now and just emerging, is this idea of drug-microbiome interactions, whereby xenobiotics or drugs which are administered like antipsychotics, as I mentioned, can change the composition, structure and function of the gut microbiota in a way that might be linked to their side effect profile, but also the idea that microbes might be able to affect drugs themselves, because microbes express a lot of enzymes which maybe can activate or deactivate drugs or affect their pharmacokinetics and pharmacodynamics, with implications for toxicity, efficacy, and bioavailability. So that's the field of pharmacomicrobiomics, which is attracting a lot of interest at the minute. So I suppose just in terms of a summary and conclusions of what I've told you, is that the gut microbiota is both stress susceptible and can regulate the stress response. It also regulates behaviors and physiology relevant to neuropsychiatric disorders. There may be an important overlap there with the capacity of the gut microbiota to regulate tryptophan availability and metabolism. And what's really going to be interesting over the next number of years is to see how well these preclinical studies translate to the clinic, and whether indeed we can develop microbial-based strategies for the treatment of stress-related disorders. So I'm just going to finish up with some acknowledgements there of the larger Brain-Gut Axis section, the Laboratory of Neurogastroenterology here. And just to bring your attention as well to the fact that World Microbiome Day is penciled in for June 7th this year. And the focus of this now annual event is going to be antibiotic resistance this year. So you should keep an eye out for activities in your area for that, or even if you're interested in producing or curating some events, we'd be delighted to hear about it. So thanks very much for your attention. I'd be happy to take any questions.